screen? Is my screen visible? Hello?
Visible, sir. Visible, sir. Visible.
Yeah, very clear, very clear. Please go ahead, Vijay.
Yeah. So I have been asked to talk on typhoid vaccine update. I would try and do the best to incorporate whatever information is available up to date. Now, typhoid, remember, is a public health problem in low middle income countries like India. If you look at the estimates, there are about 17.8 million cases occurring per year. And in India, particularly, as per the 2016 data, there were about 6.6 million cases with 66,439 deaths. It's a huge number, as good or as bad as your other vaccine preventable disease. If you look at rotaviral deaths, the estimate is around 74,000 deaths a year. And typhoid is 66,000, somewhere close to that. And we've been talking so much about rotavirus vaccine, and it is already there in the National Immunization Program, fortunately. And as I walk you through my slides, in the end, I would tell you why we need to fast track inclusion of the typhoid conjugate vaccine in the National Immunization Program. Well, there are very few community-based surveillance data study available. There is one which is underway by the Dr. Gagandeep Khan group, where 3 or 4 sites have been included, and that community data is not yet out. We are doing a community-based, hospital-based surveillance in Navi Mumbai, where there are 5 or 6 centers, including my own hospital. And there is some data which is available. It is too early to talk about the seasonality or the secular trend that we see inter or intra-year variation in the disease incidence and so on and so forth. As a part of our study, we are also vaccinating children between the age group 9 months and 15 years. And the Navi Mumbai is 22 nodes, and half of the city has already been vaccinated. 11 nodes. MR drive last year, for which we had to postpone our second phase of vaccination, and then we were hit by COVID. So the second phase of vaccination has been put on hold. Otherwise, we were really looking forward to a great impact data emerging out of this Navi Mumbai project. I'm sure in due course of time, we will be able to come out with some data which would be very helpful in making recommendations on the use of this vaccine and inclusion of this vaccine in the National Immunization Program. Hospitalized children data in our country, who are admitted with acute febrile illness, the prevalence is about 10%, and it is highest in age group 2 to 4 years. So we do see typhoid, a lot of typhoid in very young age, and the typhoid age incidence is lower in countries like India and Pakistan. Is one of the highest estimated disease burden globally. Look at the dark red color seen on this map. Only next to Pakistan. Pakistan are about 450 cases per 100,000 person years. As against that, we have about 200 plus. And remember, there is a lot of inter-country, geographic variation. So Kolkata would see about 273 plus, whereas Delhi would see 140, so on and so forth. And what is really bothering us is there's a bit of overlap with other infections which are very common in the tropics, and the typical presentation is increasingly becoming uncommon. That's happening with many other pediatric infectious diseases. And then partly we are also responsible for our irrational use of antipyretics like mefenamic acid and diclofenac, so on and so forth, which would definitely interfere with our clinical diagnosis. And then all of us are pretty well aware of the antibiotic abuse that happens. So that really poses a diagnostic difficulty. Now, if you look at the disease burden, which is recorded or documented in any surveillance system, it only represents the tip of the iceberg. You have laboratory-based surveillance, health provider-based surveillance, and population-based surveillance. It is extremely difficult to carry out a surveillance of an infectious disease like enteric fever. However, whatever data we have from the laboratory-based surveillance and what comes out in literature is only tip of the iceberg. There is a huge, huge disease burden, and which is partly, it correlates with the estimates which are given by different studies that we have talked about 17 million plus cases per annum. And one must remember that children are disproportionately affected by typhoid fever. The peak incidence between 5 and 15, I have already said that India sees typhoid in very young children. If you look at this figure, which is encircled, as early as 0 to 4 years, even children less than 2 years, you see a lot of enteric fever. The second problem that is the antibiotic resistance. If you look at the historical data and the antibiotic susceptibility of Salmonella typhi, there was pre-antibiotic era where enteric fever had a very high case fatality rate of 15 to 20%. And then came the first line of agents like chloramphenicol, ampicillin, cotrimoxazole, and we were very happy treating enteric fever, and that helped us bring down the CFR to less than 1%. And then came 1990s, where Dombivli fever, and then which was identified as multi-drug resistant typhoid fever, which spread all across the country, not only within the country, but to many other neighboring countries. The clade 58, which carried the resistance across the globe. This emergence of drug-resistant typhoid in 1990s again brought us to of despair, but fortunately, we embarked on newer agents like fluoroquinolones. Then we had third-gen cephalosporin and macrolides, which were effective in tackling this intracellular infection. But thanks to again our abuse of the fluoroquinolones, we very soon developed a lot of nalidixic acid-resistant Salmonella typhi. Nalidixic acid resistance, as we are aware, is taken as the surrogate for fluoroquinolone resistance. The MIC of Salmonella typhi or of third-gen cephalosporin against Salmonella typhi is every passing day the MIC is jumping up. And our neighboring country Pakistan documented an outbreak of ceftriaxone resistant MDR typhoid in the year 2016, and which has started spreading to neighboring countries, and they have this strain has spread as far as Europe as well. In India also, we have already documented clinical and microbiological failure with azithromycin. So imagine tomorrow if we start developing MDR typhoid, which is resistant to both the ceftriaxone and azithromycin, we'll be left with only one medication, that is carbapenem, meropenem. And then they imagine the plight of our patients who would require hospitalization and therapy with costly agents like carbapenems. And all of us are pretty well aware that the moment carbapenems are used to treat gram-negative infection, that use itself induces production of carbapenemase. So, friends, let us not take this lying down or very lightly. Enteric fever is a serious problem. And then XDR typhoid, as I was saying, this is something which is carried on plasmid. And plasmid-borne resistance easily transmits across species. In fact, Salmonella acquired it from the E. coli, from the sewage water. And that is how the XDR typhoid originated in Karachi. So this is, we have come this far as antimicrobial resistance is concerned. So this clade 15 has spread originated from India and spread to different countries. And the thickness and the length of the arrow represents the quantum of spread that it has led to. The only silver lining to the dark cloud is in many parts of our country, thanks to less use of these first-line agents like ampicillin, chloramphenicol, cotrimoxazole, there is return of sensitivity to these agents amongst the Salmonella typhi. So Salmonella typhi, the antibiotic arsenal for typhoid treatment is receding. S. typhi can transfer from MDR to XDR in a single step by acquisition of a plasmid-borne resistant gene. And XDR clone, like your MDR clone, can really spread globally. So we ought to prevent this vaccine-preventable disease. This disease, where there are so many low-cost measures, go a long way in reducing the disease burden. So what are these low-cost measures? Wash, water, sanitation, and hygiene. These have been practiced ever since we came to know of typhoid and the pathogenesis and the mode of transmission. However, improvements in water supply and sanitation in developing countries must be the preferred long-term intervention. But these are expensive and they require time to deploy, a lot of political will. And I'm not saying that we should not indulge in this. Of course, we must continue to, take these measures. But what is of importance is widespread use of typhoid vaccines can really quickly diminish the typhoid burden by modifying the susceptibility of the population at risk. And if you look at the WHO recommendation in 2018 paper, though typhoid vaccines have been made available since the year 2008, they have been used suboptimally, even in countries where the disease is endemic. So WHO recommends programmatic use of typhoid vaccine for control of typhoid fever, and all the immunization program should be implemented in the context of other efforts. So these, while we continue to use these efforts, vaccination should be, looked at as one of the most important, easy to reach masses tool to control this vaccine preventable typhoid disease. Now, having said that, let us look at typhoid vaccines. If you look at again evolution of typhoid vaccine, those senior practitioners among the viewers would recollect that there was a vaccine called TAB vaccine, which was heat inactivated phenol preserved vaccine with an efficacy of about 51 to 80% lasting for about 7 years. This was a vaccine with high reactogenicity, and people used to say that they were wondering whether to have typhoid or the vaccine. That used to be the reactogenicity. That fell in disrepute and the vaccine was discontinued. If the country wants, they can still start producing this vaccine. It continues to remain a licensed product. The oral, live vaccine derived from the Ty21A strain. It's a GAL-E mutant of Salmonella typhi. However, this strain or this type cannot synthesize VI polysaccharide. And then finally, we have the polysaccharide vaccine, which is a subunit vaccine developed from the wild type of the Salmonella typhi. And 25 micron of the antigen has been used, and the polysaccharide vaccine is still available in India, though has fallen out of use, and we no longer use this polysaccharide vaccine. It is available international, so marketed by Sanofi, and this polysaccharide vaccine is used in some developed countries. Has a variable efficacy of 55 to 72% lasting for about 3 years. And then finally, we have conjugate vaccine, where different conjugating proteins have been used, and I'll expand on this as we move along the presentation. So as I said, the TAB vaccine fell in disrepute because of the high reactogenicity, which occurred in up to 10% of the subjects. The oral vaccine is available in two forms. One is liquid to be used in children as young as 2 years, and for children above 6 years, you have capsules. And one on alternate day. This vaccine was available for a short period of time, even in our country. It induced a very strong cell-mediated immunity. However, since this strain itself is incapable of producing the VI capsule, there are no anti-VI antibodies with this vaccine. In the Chile vaccine effectiveness data, they have documented an efficacy of 53 to 78%. The problem with polysaccharide vaccine was the varying efficacy. In one of the largest field effectiveness data, which was collected and collated from Kolkata, and efficacy of 80% was documented in the age group 2 to 5 years, lasting for about 3 years. And then the same vaccine, when it was tested in Karachi, the efficacy was 38%. So varying efficacy has really bogged this vaccine. And apart from that, knowing that it's a polysaccharide vaccine, it cannot be used in children less than 2 years. It does not induce memory response. So every time you need to revaccinate as the vaccine antibodies decay. And these antibodies are of inferior quality with poor affinity. So the world was looking out for a better quality vaccine, and then the typhoid conjugate vaccine. So just to quickly recapitulate the advantages of conjugate vaccine, it induces T cells, and it's a T cell dependent vaccine, which produces very high titers of good quality IgG antibodies. It induces memory. So on re-exposure or on challenge to the same organism, you have a quick anamnestic response, preventing infection and disease in such immunized individuals. And hyporesponsiveness, a phenomenon which is pretty well known with polysaccharide vaccines, though not very markedly seen with polysaccharide typhoid vaccine. There is no risk of this hyporesponsiveness with conjugate vaccine. And the polysaccharide vaccine, all of us are pretty well aware that cannot be used in children younger than 2 years of age. And we saw in the disease epidemiology that typhoid does occur in young children. And all of us in practice, I'm sure, must have seen cases occurring in children as young as 6 months. Talking about typhoid conjugate vaccine, you have the VI polysaccharide as the source, the immunogen, which is either derived from Salmonella typhi or Citrobacter frundi. And now there is a work going on where this antigen has been extracted from fruit or plant. And then these are the different carrier proteins which have been used. So you have VI polysaccharide, which is combined with the recombinant exoprotein of Pseudomonas. This is the first experimental vaccine which was studied by the NIH of the US. It has not seen the commercial manufacturing and production, but there is an interest shown by the Chinese manufacturer, and this vaccine is likely to hit the market. And then the game changer was the TT conjugated VI polysaccharide vaccine brought by Bharat Biotech International Limited in India. At around the same time, Biomed also produced what is marketed as Pedatype. And we are well aware that Zydus has now come out with a VIPS TT conjugate vaccine, Zyvac TCV. I have highlighted this Biomed in yellow. Biomed uses 5 microgram of the polysaccharide as against 25 microgram used by other conjugate vaccines. Then there was an experimental CRM vaccine which was tried out by Novartis. They tried in 5 microgram strength, and there was a different sort of immunogenicity at different age groups. Trials were carried out in Philippines, Pakistan, and India and Bangladesh. And this technology subsequently was transferred to biological Evans. And today we have a typhoid conjugate vaccine from B. Again, I have marked this in red because apart from the source of polysaccharide being Salmonella typhi, there is yet another source called Citrobacter frundi, the VI polysaccharide from Citrobacter frundi, which is exactly identical to the one from Salmonella typhi, is used in this typhoid conjugate vaccine, which is manufactured and marketed by biological Evans, B vaccine, which is available in India now. This uses the CRM as conjugating protein. And there is a vaccine which is being developed by the IVI of Korea, where DT is used as carrier protein with 25 microgram of the VI polysaccharide antigen. Talking about the source of VI polysaccharide, C. frundi is a biosafety level 3 product where there is minimal risk of the operator and the risk of infection to the individuals who are involved in manufacturing the or working in the laboratory. As I was talking before, plant source have been identified. The pectin purified from fruit has been used to produce a molecule which is similar to your the VI polysaccharide antigen of Salmonella typhi. The molecular weight of this plant extract is low. That makes conjugation process and filtration process much easier. Now remember, let's not trying to get into the technical details of conjugation process, but suffices to remember that it is the optimum O acetylation which is important, which decides the immunogenicity. And apart from that, the other factor which is important from immunogenicity of a conjugate vaccine is the linker scheme. Fortunately, all these conjugate vaccines with different carrier protein are using the same linker scheme, and they have found to be pretty effective. Now we know that efficacy studies are something which are not so easy to carry out. So if you look at some immunologic correlate of protection, meaning thereby you give a vaccine, measure the antibody response, and then correlate that with the efficacy. And vaccines say like pneumococcal conjugate vaccine, where we know that 0.35 microgram is taken as a definite serologic correlate of protection. So unlike pneumococcal conjugate vaccine for typhoid fever, the definite correlate of protective levels of antibodies or what protects is not known for certainty. However, anti-VI IgG are taken as surrogate of protection, and the levels of 4.3 microgram per ml, which corresponds to 3.52 international unit per ELISA unit per ml, is considered as protective. This was based on the data which was analyzed from the first recombinant exoprotein of Pseudomonas vaccine, which was tried by NIH. They reanalyzed this data because these subjects which were recruited in the trial have been followed up. And what they realized that without any breakthrough disease in these subjects, when they looked at the antibody titers, it was as low as 1.2 to 2 microgram. So these now some authorities take 2 microgram per ml as the correlate of protection, an indirect surrogate of protection. So this 2 microgram is taken as a product. Why I'm talking about this 4.3 versus 2 microgram will be very clear when I pose the next question. What is the optimum dose of polysaccharide that should be included in the vaccine? So, different doses have been tried, 5, 12.5, 25. One has to strike a balance between reactogenicity and immunogenicity. And that is why optimum dose needs to be decided. When the 4.3 was taken as cutoff, if you look at the 5 microgram dose vaccine, the seroprotection came down to 77 at the end of 52 weeks, at the end of 1 year. Whereas when the cutoff was lowered to 2, as per the reanalysis done on the Pseudomonas vaccine, exoprotein vaccine, the seroprotection, 100% of the individuals continued to have levels above 2 micrograms, being thereby perhaps 5 microgram dose would also work. And many of us are those who have used the Pediatric vaccine, which utilizes 5 microgram of the polysaccharide, have seen that the vaccine works. And there is a data for about 12 months on about 1,000 subjects where there has been no breakthrough, typhoid, meaning thereby 100% efficacy over a short period of time. These subjects are now being followed up, and we have an unpublished data of 2.5 years. Now, what made the WHO recommend the vaccine was the immunogenicity data submitted by the Bharat Biotech vaccine, type bar TCV. Large number of subjects were recruited in trials which had 2 limbs. One children between 6 months to 2 years, and the other was 2 months to 45 years. 6 months to 2 years obviously did not have a comparator polysaccharide arm, because polysaccharide vaccine was not recommended, is not recommended for use below the age of 2 years, whereas these older children have a comparator of polysaccharide vaccine. And some subset of these children underwent boosting at 2 years, some did not receive any booster. And that gave a lot of information. And I would not bore you with the statistical data, but suffices to say that it demonstrated persistence of GMCs, antibody geometric mean titers, well above the minimum which is required for protection. Though the antibody titers dipped, they remained above the minimum required for protection in non-boosted cohort as well as in boosted cohort. Of course, booster cohort demonstrated excellent boostability. And if you look at the seroconversion, percentage of individuals who remain seroconverted, again, there was a beautiful response seen. So when you study a vaccine, you look at one geometric mean concentration of antibodies, that is antibody titer, and the seroconversion, where you look at the percentage of population who remain seroprotected. The second boost came when this type bar TCV underwent what is known as human challenge study in Oxford, done by Dr. Andrew Pollard, where human beings were vaccinated. Of course, these belong to area where typhoid is not endemic. They were given single dose of typhoid conjugate, and the other arm received typhoid polysaccharide vaccine. And they were then orally challenged with the Salmonella typhi bacteria. And this study, the conclusion was it demonstrated an excellent efficacy of 87.1% with the typhoid conjugate vaccine. And then there was a analysis of data where what they call a sero efficacy study. I'll explain you what it means. That when you inject an individual with a vaccine, you induce antibody production, and these antibodies decay over a period of time. And if the antibodies which are expected to decline, if there is sudden increase in antibody levels, or if there is a sharp decline when the antibodies are expected to plateau, that indirectly indicates that this individual has been challenged by the same pathogen, meaning thereby he has been exposed to the pathogen and he has suffered subclinical infection. And with this methodology, they could derive a sero efficacy of 85%. So human challenge 87%, sero efficacy data giving efficacy of 85%. This made the WHO to make a strong recommendation of this typhoid conjugate vaccine, and for children as young as 6 months of age. So they made recommendations for one single dose. So this is what the WHO position paper read in May 2018, that TCV is preferred in all age groups, a single dose of 0.5 ml in children from 6 months, and in adults up to 45 years in endemic region. And what they hinted at was the timing when the child comes for measles containing vaccine at 9 months would be optimum time to give this vaccine. The Pseudomonas vaccine demonstrated immunointerference when administered with measles So there was a recommendation of separating these two vaccines by a 4 weeks interval. However, Bharat Biotech went ahead and did a study and where they documented that there is no immunointerference and these vaccines could be given together. And catch-up vaccine has been recommended by WHO. The same was adopted by the ACVIP. ACVIP also went on to recommend single dose of typhoid conjugate vaccine. Now, what is the matter of discussion in many fora and amongst many pediatricians is the number of doses. What is the optimum number of doses required for good protection? Well, the answer is it is not known because we have so many limitations. However, if you look at the data, efficacy data of the recombinant exoprotein of Pseudomonas, at the end of 4 years, one dose demonstrated efficacy of 87%. And in school children up to 8 years who were followed up, and adult up to 10 years, they had good efficacy, even with some single dose in some subjects. Pedatype, the Indian brand which used 5 microgram and TTS conjugating protein, as I said, demonstrated 100% efficacy. And 62 subjects of this small cohort had 100% seroconversion at 6 weeks with single dose. There is a post-marketing data available from Chennai, where they demonstrated 80% seroconversion after single dose of Pedatype. However, if you closely look at the antibody titers, kinetics in subjects who did not receive booster in the Bharat Biotech trial, what you see in the red, in the open label trial in young children, there is a dip and then there is a increase in the GMCs and also increase in the seroconversion. So how did this happen in the absence of a booster? Probably because of natural subclinical infection. So can one really rely on a natural infection? And of course, those who received booster demonstrated excellent boostability. Both the seroconversion and the GMCs rose very rapidly. There is an unpublished data from Bharat Biotech, which looks at the 7-year data. And in unboosted subjects, the GMTs were 54 versus 101 in boosted. And the seroconversion dropped to 45 to 60 as compared to 80 in boosted subjects. So there is a dip in seroconversion. And then there is something called avidity index, which indirectly talks about the quality of antibody. Higher affinity antibodies have very high avidity index. In unboosted, the percentage of individuals in the unboosted rim was very small, who had avidity index of more than 60, whereas in boosted it was very high. So if you put all this information together, there is suboptimal immune response in children below the age of 2 years because of the absence of the immunologic edifices, which are responsible for exposing the antigen to the antibody production house, that is lymphoid tissue. There is a significant fall in seroconversion to as low as 60%, we saw from that table in unboosted individuals. GMCs and seroconversion rise subsequently in both these boosted and unboosted. And in unboosted, it perhaps rose because of the subclinical infection. And this natural boosting cannot be relied upon in very young children, especially when it comes to offering the best kind of protection in your private practice. Quality of the antibodies may not be as good. We looked at the avidity index. The percentage of individuals who had avidity index of more than 60 was low in unboosted group. And there is a good boostability which has been demonstrated in subjects who received booster. So considering all this, I personally feel that there is a need to have 2 doses. Remember, typhoid is an inoculum-based bacterial illness. Bacterial antigens somehow are poor immunogen as compared to a viral antigen. So I personally feel that a second dose of typhoid conjugate vaccine is required. What about safety of this vaccine? There is a huge data from the post-marketing surveillance. 5,000 plus PMS forms have been evaluated and it has stood the test of time. There is a global advisory committee on vaccine safety which has cleared this vaccine, and they recommended that in countries or in places where the vaccine will be used, they must look for the surveillance. And in Navi Mumbai, we conducted this, we had the opportunity of looking at the safety on more than 1 lakh children who received the vaccine in the first phase. We did telephonic follow-up. We used the standard AI5 machinery that is in place at the Navi Mumbai Municipal Corporation Hospital. And we also carried out hospital-based surveillance using the Brighton collaboration definition. This is not the platform to elaborate on what it means, but suffices to say these are the parameters on which the vaccine safety is assessed. We injected about 113,420 doses to children from Navi Mumbai, And what the study concluded, this has already been published in international journal, that major of the events reported AFI surveillance and active phone follow-ups were mild and occurred within 5 days of vaccination. Occurrence of adverse events of special interest in the unvaccinated population was higher than in vaccinated population. There were cases of thrombocytopenia because it coincided with the dengue season in our Mumbai. And for this generated further evidence of excellent safety profile of type bar TCV when administered to children from between 9 months and 15 years of age. What about cost effectiveness? There is a study which talks about that for every true case of typhoid fever, there are about 25 cases of acute febrile illness which are non-typhoid but treated as typhoid under admission and antibiotic use. So it is a cost-effective proposition to use typhoid vaccine. There are some limitations of this conjugate vaccine, lack of large-scale efficacy or effectiveness data, which is likely to come from areas where this vaccine is put to use. Let me tell you that Pakistan, Bangladesh, Nepal, and Malawi are other countries where this vaccine has been exported and has been put to use. There is no effectiveness against paratyphy, non-typhoidal salmonella. We know that NTS incidence in one of the surveys done in Kolkata is 1.8 per 100,000 population. These vaccines are also not very effective in VI negative serotypes. So Nepal, Bangladesh, and Malawi and Navi Mumbai study is likely to throw some more light on the vaccine efficacy. Nepal efficacy is already out and the preliminary data shows an efficacy of 81.6%. Now, this is the second last slide where I have talked about there are so many vaccines which are in different stages of production. What I would draw your attention to is the Pseudomonas exoprotein vaccine, which is being manufactured in China, and it is, they have shown interest in WHO pre-qualification. And this is something which would really be, one of very good vaccine which has already undergone efficacy data. And finally, I would say what is there in store in future is what we look at is subunit vaccine or novel linking methods which would improve the immunogenicity and longevity of protection, use of carrier protein as antigen. There are 3 oral vaccines which are derived from TY2 strains, which are much stable than TY21 strains, and there are under different stages of development. This oral vaccine again would be a great boon. So friends, to conclude, typhoid is a public health problem in low middle income countries. Effective conjugate vaccines are now available. While we continue to practice WASH, that is water, sanitation, and hygiene, vaccine can reach masses and help prevent typhoid. There is a need for newer generation serotype independent vaccines which are effective in salmonella typhi, paratyphi, and non-typhoidal salmonella. Typhoid is a vaccine preventable public health problem, and I'm sure you will be convinced that with the evidence of the burden of disease and availability of an efficacious, safe, and cost-effective vaccine, which is very much feasible to implement, we definitely need to fast-track this inclusion of typhoid conjugate vaccine in our national immunization program. Thank you so much. If there are questions, I'll be more than happy to take them up.
Thank you, Vijay, for that brilliant century, chanceless century, as I said earlier. Yeah. But let us take the questions. One of the questions Ashwath has asked, interestingly, is that typhoid is a disease of children above 5 years. Why does the IAP recommend vaccination from 6 months onwards? What's your take on it?
My straightforward take is, Dr. Bala would also agree with me that there is enough evidence to say that typhoid does occur in children as young as 6 months. You have about 10% of typhoid cases occurring in children less than 2 years of age. And we know that, and the most affected age would be children between 5 and 15, but there is a significant burden in children less than 5 years of age, and we see about 10% of cases in children less than 2 years. We have taken up active surveillance in Navi Mumbai at 6 different hospitals, where every patient of AFI undergoes blood culture examination, and we have documented a similar kind of finding over last 2 years. 10% of our typhoid has occurred in children younger than 2 years of age.
Somebody has asked, paratyphoid is on the rise. Is there going to be a vaccination for it?
Paratyphoid vaccination is under development. Bharat Biotech, BE, and there are several other manufacturers who are looking at a combination vaccine of typhoid and paratyphoid. This paratyphoid, rise in paratyphoid could be a relative rise. We have started using a vaccine which is specific for Salmonella typhi, and that's the reason why the burden of there is a relative increase in the paratyphoid burden. But however, in the absence of a vaccine against paratyphoid, let us continue to use at least one which is available, which can prevent a huge disease burden. 66,000 deaths, as good or as bad as your rotaviral deaths.
Jagdish has responded by saying, great talk as usual. I think everybody would agree. But he has also mentioned that we do not have convincing data for second dose. Just going by antibody decay is insufficient. And what is the incidence of typhoid in vaccinated children? He says, in my experience, I have had no typhoid disease in over 500 pediatrics. Your response, Vijay?
Yeah, I totally agree with him that there is no robust evidence to say that a second dose of typhoid is required. However, there is no evidence to say that one dose would offer protection in long run. We do not know definite correlate of protection. And yes, we do see typhoid, breakthrough typhoid in vaccinated individuals. In the Navi Mumbai 2-year data, we have seen at my own center about 3 or 4 cases out of 80 cases who have had typhoid following 2 doses of vaccine.
Dr. Vijay, I would say that, unless we have definite data, see, most of the opinions for the second dose are all personal opinions.
Yeah, I totally agree with you.
Yeah, let me complete. Most, I would say why most, 100% of the consideration for second dose has been on personal opinion and not on immunological evidence, because we do not know what is the protective. It's like, antibody titers following COVID vaccination. So it is unfortunate that in a country like ours, where we are using typhoid vaccine, we still have not generated enough data to sort out this problem. It is time that we really come out with data, whether 1 dose is good, 2 doses. Somebody might say even 3 doses may be required. What is the right one? We need answers for it. And then one more.
I cannot agree more on Bala. I cannot agree more on what you say. However, that is what I'm saying. I'm not saying that ACVIP needs to have a very strong data to make change in the recommendation. I totally agree, and I respect the ACVIP.
Incidentally, last month I saw a child who had typhoid after 2 doses of typhoid vaccine. Okay,
We have
so
seen,
that's not enough evidence to say 3rd dose is required.
absolutely. I agree with.
Giving a personal opinion, saying 3rd dose is required. That's one. Last question. Somebody has asked, are they using the same antigen in the polysaccharide vaccine for conjugation, or they are using a different antigen?
No, same antigen. The source is different. In the BE vaccine, it is Citrobacter frundi. In other, it is the wild type of S. Salmonella typhi. And the Pedatype uses 5 microgram, where all other vaccines are using 25 microgram. The Novartis vaccine, in the trial, they use 5 microgram, but the B chose to use 25, retain 25 microgram as the strength.
Okay. Vijay, one question. Should all the pediatricians take typhoid vaccine, conjugate vaccine, regularly, any age? What would you recommend?
Again, there is no evidence to say, document the immunogenicity and efficacy, but I would say all of us must take at least 1 dose of conjugate vaccine.
Okay, what
Since
about...
there are trials carried out at the age of 45 years, there is no harm in taking this vaccine.
Somebody has asked, what about those who have received polysaccharide vaccine earlier? Do you give vaccine? The answer is yes.
See, for polysaccharide vaccine, the dose was revaccination with yet another dose of polysaccharide every 3 years. Now that a conjugate vaccine is available, go ahead and vaccinate them with single dose of conjugate vaccine.
Okay, it's exactly 8:30 p.m. Thank you, Vijay Vele, for finishing in
One
time.
question, sir.
Over to Dr. Rajendran.
Dr. Rajendran wants to ask something.